one is about a special type of mechanism seen in plant that is called apomixis. The term is of apomixis. It is the production of seed without fertilization. Initially you have studied it is a product of sexual reproduction, but apomixis is a production of seed without fertilization. It is an asexual mode of reproduction, but it imitates or mimics the sexual reproduction. So, how the apomixed seeds are formed? For example, in case of grass as well as in the family Asteraceae, in case of grass as well as in the family Asteraceae, normally the egg mother cell is always diploid in nature because angiospermic plant is diploid, so the egg mother cell is also diploid. This egg mother cell, without undergoing meiosis, directly it forms the egg cell. So egg mother cell is of diploid. Without undergoing meiosis or reduction division, it is forming the egg cell. So what will happen? Egg cell is also diploid. This diploid egg cell can directly form the embryo. In case of grass and Asteraceae, as the angiospermic plant is of diploid, egg mother cell is diploid. Without undergoing meiotic cell division, it forms a diploid egg. This diploid egg directly forms the embryo. But in case of citrus and mango, so for example, let us see here, this is the structure of a ovule. So in case of citrus and mango, what is happening? This cell is called the nucellar cell. So some of the nucellar cells will enter into the embryo sac. Some of the nucellar cell will enter into the embryo sac and that forms the embryo. So this is how which seed is formed? Apomixed seeds are produced. So what is apomixis? Seed production without fertilization. So here it is an asexual mode of reproduction, but it mimics the sexual mode. In case of grass and Asteraceae, egg mother cell is of diploid. Without meiosis, directly it forms the egg cell and that forms the embryo. But in case of citrus and mango, the nucellar cell surrounding the embryo sac enter into the embryo sac and form the embryo. This is how apomixed seeds are formed, and it is a very important question. Now what are the uses of this apomixed seeds? One advantage is that it is cheaper than hybrid seeds. So yesterday you have studied about, day before yesterday you have studied artificial hybridization. So when you compare to the hybrid seeds, apomixed seeds are very cheaper. Second advantage is, for example, when you combine the desirable character present in two different plants into a single one, then the process is called as hybridization. Product is called a hybrid. But actually when we take this hybrid seed to the field, there what will happen? The pollen of any undesirable plant also can reach the stigma. That time whatever good qualities we have combined can be separated or segregated. But in case of an apomixed seed, once the characters are combined, it cannot be segregated. Clear? In case of hybrid, we are combining the character. But when we take it to the actual field, there are possibilities for undesirable pollen to reach the stigma. That time what will happen? Whatever characters we combine, it gets separated or segregated. But in case of apomixed seeds, once the characters are combined, it cannot be segregated. That is the advantage of apomixed seed. So now what is going on? Research work is going on to transfer the apomixed gene into the hybrid variety so that farmers can keep on using the hybrid seed year after year. But there is a disadvantage is also there in apomixed seed. What is the disadvantage? As it is formed without fertilization, there is no genetic variation. In case of apomixed seeds, there is no genetic variation. So apomixed seeds are having both advantages and disadvantage. Advantages are it is cheaper than hybrid seeds. Once the characters are combined, it cannot be separated. But the disadvantage is as it is formed without fertilization, there is no genetic variation. This is clear. It is a very important question. Now next phenomenon is called as polyembryony. So you know very well that the term poly refers to many. Many embryo in a seed is called polyembryony. Presence of many embryo in a single seed is called as polyembryony. So this you can see now. Nowadays in our house when we are using orange or lemon, take the seed, just open it out and see many small bulb like structures you can see. That is what is called as polyembryony. Presence of many embryo in a single seed. Example is of orange, lemon, etc. So how this polyembryony occur? Just now you have studied when in case of citrus and mango, the nucellar cell enter into the embryo sac and form the embryo. So when many nucellar cells enter into the embryo sac, every one can form a embryo or a seed. So this is how many embryos are formed in a seed. And another thing is here also the disadvantage is that there is no genetic variation. Here polyembryony, presence of many embryo in a seed, commonly seen in case of citrus varieties like orange and lemon. Inside the seed we can see the presence of many embryo. How it is formed? The nucellar cell surrounding the embryo sac enter into the embryo sac and form the embryo. But here the disadvantage is that there is no genetic variation. And then when you have studied about the pollen grain is having certain advantages. What are all the advantages it is having? One advantage is that it is rich in case of nutrient content. So people are using it as a food supplement. Athletes are using as a food supplement to improve their performance. They are consuming it to enrich their energy level. Now the pollen grains are available either in the form of a pollen tablet or syrup in the market. So that is the advantage of pollen grain. At the same time, what is the disadvantage of pollen grain? For example, the pollen grains of some species like Parthenium, or commonly we call it as carrot grass, that causes allergic type of reaction like asthma or bronchitis. So when you inhale the pollen grain of Parthenium or carrot grass, in some people it causes allergic reactions like asthma as well as bronchitis. So pollen grain is having both advantages as well as the disadvantages. Now if you see the viability or fertility of the pollen grain, in some the viability depends on temperature and humidity. In some cereals like rice and wheat, pollen grain lose its viability within 30 minutes time. Once the anther releases the pollen, it remains in the viable condition or in the fertile condition only for 30 minutes duration. But in case of Rosaceae, Leguminaceae and Solanaceae family, the pollen grain remains viable for many months even. But the viability of the pollen mainly depends on two factors. One is of temperature as well as humidity. Even then also in rice and wheat it is viable only for 30 minutes. But in Rosaceae, Solanaceae and Leguminaceae it remains viable for many months. Next term is about cryopreservation. It is the preservation of pollen grain. So how to preserve the pollen grain? Pollen grains can be preserved in liquid nitrogen at minus 6 degree Celsius, at minus 196 degrees Celsius. That method is called cryopreservation. Now the question arises, why to preserve this pollen grain? The important thing to preserve the pollen grain is that, for example, if the plant is the last member of the species, when this plant died, the entire species will become extinct. In that cases, when we preserve the pollen grain, it can, in a pollen bank, we can use it for continuation of its species. So this method comes under ex situ method of conservation. So cryopreservation is the preservation of pollen grain in liquid nitrogen at minus 196 degrees Celsius. It is preserved in pollen bank for future or to maintain the continuity of a species as well as for plant breeding programs. So today you have studied what is a seed, what is the structure of a seed, significance of a seed, and then comes apomixis seeds, advantages and disadvantages, polyembryony and its disadvantage, then pollen viability, preservation of pollen grains. Thank you all. If you have any doubt, very frankly clarify it through your WhatsApp group. Thank you, children.